What is up, my amazing people? Okay, genetic technology. Here we go. Let's knock this out. For this unit, again, everything is like all over the place. If you want to pause the video, look at all these things, maybe draw some connections, concept maps, huge. They're really good for like making your own personal connections. That's some education stuff. Did a thing, mostly a thing for like Morgan and Jada and Madison. Oh, I did a thing. This little smiley face means it's probably one you need to pay attention to because there's a lot of like examples and stuff that you won't necessarily see. They're good to look at and like be able to apply, like, oh, how is this actual like biotechnology or concept found in the world, and like how could I personally apply it, and here's an application of it. But don't feel like you need to go and memorize all these examples, but you know how my tests work at this point. I'm going to give you a lot of examples in this one, and you're not necessarily going to see them, but you need to know kind of the application of the technology or the concept that we're talking about. So biotech is our big concept, and that's all this one is going to focus on. It's just basically the process of manipulating DNA. And so then we can use those for different like medicines. We can use them for, study of disease. We can use them for a lot of different things. So the big ones that we talked about: cloned animals, GMOs, transgenic animals, and then obviously pharmaceuticals is a really big one, and gel electrophoresis. And these two kind of play hand in hand, and these kind of all play hand in hand. So I'm going to get to here today, and then I'll save the next video. I say today, but I'm literally going to go film the next one right after this. So gel electrophoresis I'll do on the next video. So just manipulating DNA, all the different uses of them. So let's talk about cloned animals first. The biggest thing with a cloned animal is we are making a genetically identical organism. Now, when we say genetically identical organism, we do that by taking an egg, which is haploid, and removing all of the DNA from it. So it's just an empty egg. It does have all of the cytoplasmic determinants in it, and that's the stuff that gets the embryo dividing and going through mitosis. And it does have all of the, mitochondrial DNA that it would need. So it's like got all the stuff to be a functioning egg. It just doesn't have its DNA in it anymore. And what we're going to do is we're going to take DNA from a donor, from a somatic cell. We usually want to do this in people. When we talk about it being done for people, it's really not cloning the entire organism. It's so that we can reproduce a lot of stem cells. And we'll get to those in a second. So anyway, we steal all of that DNA, we put it in the egg, and now the egg thinks it's diploid. So it thinks it's fertilized. So it has all of the DNA it needs to go off and be a zygote, to go off and then start cellular division to become an embryo and become a fetus, and so on and so on and so on. So the long story short is it's this really crazy thing where we make a genetically identical copy. Now, when we talk about stem cells, there's a couple different words that we like to use. The first one is a stem cell is something that we like to say is pluripotent. It has a lot of different options. Potent, it's able to like be whatever it needs to be. And so a stem cell is just a baby cell that can differentiate. And so it doesn't have necessarily the chemicals in it or the right enzymes in it to be specific cells yet. It's going to have to go and make those. And so stem cells are as far as medical applications are concerned, they are super awesome because they allow us to go plug and chug those cells wherever we need them. And we've gotten really good at differentiating the cells instead of like letting them be whatever they need to be. So that's really awesome. Now, the reason like cloning got so big is because stem cells are less and less potent. They can be less and less things the farther down the developmental chain they get. So when it is like a brand new zygote, those stem cells, those cells, they can literally be anything. But as we get down the line, what you'll see when we learn about evolution is there's cells that start to migrate, and then all of a sudden the limitations that they have is more. So like now, because they've migrated this way, they can only be part of the head region. So we just cut off half of the cells that they potentially could be. And so that's kind of why cloning got really big, is because we were getting stem cells at some of their earliest age, which was really good for the potency of the cell. Okay. So here's that process. We're going to take donor cells. These are somatic cells usually. Here we took a mammary cell. It's a diploid cell. We took an egg. We inoculated it. That's a funny word, inoculated. You might see that. And then we stuck the diploid number in there. So this nucleus is diploid, and right now this egg thinks that it's fertilized. So it's going to grow and develop into an embryo. Once we get to this stage, we can put it in a surrogate mother, and all of a sudden we have Dolly the lamb. Dolly Parton, amazing. She's a great human being. Has nothing to do with this lamb. Just wanted you to know that I really like that lady. Okay, so genetic engineering also kind of has other implications. So some of the other things that we can do with it is we can take DNA and manipulate it to put two different sources of DNA together. We call this recombinant DNA. It's pretty interesting because we also call crossing over recombinant DNA. So that is annoying that they use the same word for two different things. But in this particular thing, what we're doing is we're taking DNA and we are splicing. That's a fun word. We're splicing two sources of DNA to create a different organism. That's how we make recombinant DNA. We do that with restriction enzymes. They're super important across lots of disciplines of biotech. And then obviously we can do a simple process of gene cloning, which kind of requires us to cut out certain parts or cleave certain parts of DNA and just make a bunch of copies of that gene. Usually we use a bacteria for this, and usually this is what we talk about when we talk about pharmaceuticals. So let's start looking at it. All right, let's look at restriction enzymes, just what they do. A restriction enzyme, it's responsible for cutting DNA. Two big things about the cut is, one, it is a specific enzyme cuts a specific code of DNA. So we've got, we'll use GGG. Every single time we see GGG, we're going to cut in this little zigzag between those. And that is going to be where our cleave happens. So when we cleave the DNA, we cut it so that it has these like lopsided sticky ends is what they're called. And believe it or not, like that's something that will get you points on an essay, which again, really scientific there. So these sticky ends are the purpose of, like, if you look at this, it's like you've got this lopsided thing so that we can actually put it together and ensure that we're putting stuff in the right place. So if we use the same enzyme to cut DNA out, and we use the same enzyme to cut DNA apart, when we insert the new DNA, it like fits really nicely. So that's kind of how that works. And so we actually then use our ligase, which we learned about in DNA replication, like splice that bad boy back together. So restriction enzymes, they cut it in a specific location. The sticky ends and complementary base pairing is what ensures the correct placement of inserted genes, the correct, putting together of interesting DNA. And so we can take those restriction fragments and connect them back with a ligase. Now, why is that so important? One of the best things we have going for us is all of these like pharmaceutical advancements because of it. Any IN word, insulin, any hormone, all of those like IN suffixes usually refer to like something that your body needs. That's usually a protein, and a lot of times it can be in. Tori just walked in. She left after our conversation. So we can actually get bacteria to make those for us by opening up some bacteria, inserting that DNA, and then bacteria, it grows like a freaking mad thing. It's like reproduce. And when it starts doing that, it's also using all the same mechanisms because it is a common conserved core process that DNA transcribes into mRNA, and mRNA translates into proteins. So we're actually hijacking this bacteria to do our dirty work for us, and we're going to use all of its mechanisms to make larger amounts of these different proteins, these suffix in, all of them, insulin, all the hormones that have that in them. And so that's kind of what we use it for. We can also use it to manufacture vaccines. The reason we would manufacture vaccines is because, hey, guess what a vaccine is? It's DNA that a virus has. Well, we're going to take a vaccine and we're going to manipulate it just a little bit so our body can get over it and make us susceptible to that virus. So these are all kind of like different things that we hijack this bacteria and use it for. So this is probably the coolest one for sure. And so here's what that looks like. This is a plasmid, remember? We cut that plasmid, we opened it up. We also cut out a specific code of DNA that produces. So we're going to take the human insulin code, we're going to stick it in that bacteria, and when that bacteria starts to multiply, starts to produce proteins, it's actually making human insulin for us. I'm going to click really fast. These don't have smiley faces on them. There's just a ton of examples. Okay. So we can use this restriction enzyme to make GMOs. Okay. So we've talked about the bacteria. We can also modify foods. What we'll do is we'll take things that already exist in nature. So for instance, pest resistant. Mustard is really pest resistant. So we can take mustard seed and put that pest resistant gene in other foods so that pests aren't eating them. So there's lots of benefits to GMOs just across the board. Just be wary of the fact that, like, when you're doing this, like, for lack of a better word, like playing God mentality, you don't know like what the actual implications are. So that's one big thing that they like to talk about. College Board has, like, what are the problems with this? Like, maybe it can grow uncontrollably and disrupt an ecosystem. Maybe we're making super weeds. Maybe we're making super pests because of this. And then last but not least, transgenic organisms. We're doing the same thing, but we're doing it in animals. So Farm has used this. What we'll do is we'll take, same concept with the bacteria. We'll take plants or animals to produce us different insulins or medicines, and that's just taking the human gene and putting it in so that they're also making it. One of the better examples that we have are goats. Spider silk is like very, very like, oh my gosh, make silk. Everybody wants it. Well, it's really, really long process to collect that much spider web. And so what we've actually done is we've modified goat's milk so that they're shooting out along with their milk, like webbing, like spider webs, so that they can collect that and then use it for making silk. So that is the first one because I'm going to run out of time, but you guys are awesome. You can go on to the next video. Bye.